Welcome back, Bio 153 students. And today we're going to go over Chapter 21, Blood Vessel Anatomy and Physiology, Part 2. So the second half of this chapter, the big focus is understanding the physiology. So here we go. Time to have some fun. All right, so some very important terms here to be aware of as we go through the physiology in here today. Number one is understanding the term blood flow. Blood flow is measured in units of milliliters per minute. And this is looking at the volume of blood flow, the volume of blood flowing through a blood vessel, perhaps an organ like the heart, or the entire circulation. So definitely a very important term to know as we go forward today. Number two, blood pressure. And blood pressure is measured in units of millimeters of mercury. And this is looking at the force per unit area that is applied on the wall of a blood vessel by the blood. So a very important concept to understand, blood pressure. And number three, the term resistance, or maybe just focusing on peripheral resistance. And this is looking at ultimately the opposition to the flow, the blood flow, and ultimately the measure of the amount of friction that blood may encounter. And terms that kind of correspond with the idea of resistance would be blood viscosity. And viscosity, maybe that's a new term for you. Viscosity refers to the overall thickness. So it refers to the thickness of the blood. So is the blood really sluggish, which usually means the viscosity is pretty high, or is the viscosity really low? And so then blood can flow a little faster. The other terms that you can see here would be looking at the length of the blood vessel, how long is it? And likewise, the overall diameter of the blood vessel. So these are three really important types of concepts that really correspond to understanding resistance. With that, we now take a look at overall atherosclerosis. And when we look at atherosclerosis, this is a type of what's known as arteriosclerosis, really looking at the overall walls of arteries. Maybe they're getting thick, stiffer. Typically, arteries are really prone to that, especially ones that are really close to the heart. So the aorta, your coronary arteries right and left, and the common carotid arteries right and left are typically prone to this happening. So what we're doing here is looking at an analysis, really a comparison between a human artery, such as this image shows here, versus a clogged pipe. And we can see here with this artery that you have a lot of plaque building up. That's the yellow here, the plaque. Okay, the plaque. So you can see that here. There's that plaque building up. Typically, plaque buildup is typically from deposits of lipids, triglycerides, for example. And this can come about from many, many years of living a sedentary lifestyle, not getting that blood flowing because you're not exercising, a very poor diet, and maybe eating more processed foods than fruits and vegetables, and a well-balanced diet. But other things that can lead to this as well, the significant plaque buildup. Other things would be smoking is a big risk factor. Unfortunately, even stress. Stress can definitely lead to a lot of plaque buildup as well. So we compare this idea to a clogged pipe. Maybe you've had that before at your house. So we answered this question here based off looking at this image of the artery. Would blood flow be increased or decreased? So just think about that for a moment. What are your thoughts on that? I hope you decided to put down for the answer here that the blood flow certainly would be decreased, right? Blood flow is being backed up. So this plaque, it could eventually lead to the artery being totally clotted, and then that could lead to some major issues with the heart attack, things like that. So it's very possible that maybe a stent needs to be put in, which is kind of a mesh that can be applied on that plaque to open up that vessel. So with this understanding, we now take a look at a very important law when it comes to blood flow, and we are looking at the law here, which is known as the overall Pozzoli's law. So the Pozzoli's law. This is a French term, the Pozzoli's law. And when we look at the overall Pozzoli's law, we look at the volume of blood passing per unit time through a capillary tube is directly proportional to the pressure difference between the ends of the blood vessel to the fourth power of the internal radius, and inversely proportional to the length of the blood vessel and the viscosity of the fluid. So the fluid, in this case, looking at the blood. So that probably sounds like a mouthful. You're probably thinking, whoa, what do we got to learn here today? So if you don't like this law, you can go ahead and blame it on this gentleman here, this French cardiologist that developed the overall Pozzoli's law. And so the Pozzoli's law was ultimately discovered by this gentleman here. You can see he lived out his lifetime, more in the 19th century. So overall, John Pozzoli. Okay, so John Pozzoli developed this in the 19th century. So, all right. So here we can see what the terms mean here for the overall Pozzoli's law. And we can see here that the Q is equal to the rate of blood flow. P is equal to pressure differences. So this P1 minus P2, pressure differences between the ends of the blood vessel. R refers to the radius of the blood vessel. L refers to the length of the blood vessel. And N refers to the viscosity of the blood. How thick is the blood? Continuing to look at this law so we have a good understanding of it. Again, we look at the overall Pozzoli's law. And a couple things. First, the idea that blood flow is directly proportional to the blood pressure and the blood vessel radius. While looking at blood flow can also be inversely proportional to the blood viscosity and the blood vessel length. And we can kind of see this idea here with the diagram here, what these terms might represent. And you might see delta P here. This delta refers to ultimately change. That's what that overall term is relating to when we think of a math concept. And many of you have to take Math 117, which is important when you go into the allied health field. And you probably go through some of these terms. So we can see here L referring to the length of the blood vessel. Again, delta here is referring to change, this case change in blood pressure of the blood vessel. And here we can see radius right here. All right, so let's review this law. I want to make sure we have a good understanding of how to use ultimately the Poiseuille's law here. So here we have the equation. We have Q equals P1 minus P2 in parentheses pi r to the fourth power is divided by 8 the L and eta. What do these terms mean, right? Well, Q is ultimately referring to the blood flow. So Q is referring to blood flow. How fast is the blood moving through a blood vessel? P1 minus P2 ultimately is looking at the overall change. So change when it comes to blood pressure. So the change in blood pressure in the blood vessel. So maybe we're looking at a artery, maybe, perhaps. So that's what P1 minus P2 is looking at here. The R ultimately is looking at the radius. So if we were to draw a blood vessel, so maybe we have a blood vessel here. Maybe this is the, well, I don't know. Let's say it's the hepatic artery, perhaps. There we go. The radius would be if you go from one side of the blood vessel right to the middle. So this area here would be defined as the radius right there. So that would be the radius. And we can fix that here for you. There we go. So ultimately this is the radius right there. If you went all the way across, then we're talking diameter. L is ultimately referring to the length. So how long is this blood vessel? So this is referring to length. And it looks like an N, but actually it's a Greek notation, eta. Eta is referring to the viscosity of the blood. So eta is referring to the viscosity. Okay, so eta is referring to the viscosity. So how do we put this together, right? Well, let's say we're looking at a situation where we're looking at blood flow that is proportional. Okay, so when blood flow is proportional, this idea of proportional, and got all of my handwriting here. When we look at the idea of the blood flow being proportional, in this case, directly proportional, I like to represent it by arrows. That seems to always help. So if there's a change in one variable in the equation, the net result is that blood flow will go up. Likewise, if there's a change in one variable in the equation, the net result is that blood flow will go down. So increase, increase, decrease, decrease. For example, let's say in the blood vessel that blood pressure, BP is going to stand for blood pressure. Let's say that blood pressure increases. The net result of the increase in blood pressure in that particular blood vessel, maybe it's the hepatic artery, that's ultimately going to lead to an increase in blood flow. That's definitely the case if you think of somebody that's possibly exercising, right? Your blood pressure goes up when you exercise, and blood flow will increase through an artery. So definitely the case if you think of blood flow going through the heart, it's going up through the aorta. Blood pressure is going up, it's going to increase in the body, so blood flow is going to go and increase in the aorta. Likewise, if we're looking at radius, let's say we use this little r for radius. If the radius is going to increase in that blood vessel, the net result is that the blood flow will also increase. And that makes sense. If you have more room in the blood vessel, blood flow can increase. You have more room. So that's the concept of directly proportional. Now let's go ahead and take a look at the concept of inversely proportional. So we've looked at directly proportional. Now we're going to take a look at the concept of inversely proportional. Okay, so inversely proportional. So inversely proportional and again, not the best handwriting in the world, but you get the idea. So what happens here then? Inversely proportional is when one variable goes up in the equation, the net results that the other variable decreases, in that case blood flow. And one thing to consider too, with directly proportional, it's referring to the variables on the top of the equation. Inversely proportional is referring to the variables on the bottom of the equation. So with inversely proportional, let's go ahead and we can pick on viscosity. So V for viscosity, again referring to thickness of the blood. So if the viscosity increases in the blood, the net result is that blood flow is going to go ahead and decline. Okay? Blood flow is going to go down. And this makes sense, okay? If the blood's getting a lot more thicker, sluggish, a lot harder to move in the vessel, blood flow will decline. Likewise, if the overall length of the blood vessel increases, so length here. So if the length of the blood vessel increases, the net result is that if you increase the length, eventually that blood flow will decline. Okay? That's something to consider with the aorta. In the beginning of the aorta, okay, the blood flow is moving pretty fast. But eventually, as you get towards the end of the aorta, there's many branches going off and things, and so eventually that blood flow will decline, especially with the aorta with distance from the heart. So those are considered inversely proportional components there. All right, so definitely make sure to review this. Very, very key to know this for the exam. And the thing that's here too is this is like the idea of critical thinking. Critical thinking skills are very key if you're going to go into the medical field. So critical thinking is really, really important when you go into nursing and other allied health fields. And this is looking at blood flow here. We're doing critical thinking right now. So let's go ahead and review this overall law of blood flow, looking again at Poiseuille's law. So according to Poiseuille's law, blood flow will decrease if, which of these would be true? So here we're really thinking of critical thinking skills here. According to Poiseuille's law, blood flow will decrease if, which of these would be true? All right, the correct answer here, I would recommend to go with choice letter D. So letter D is the correct answer, all of the above. So this gives you an idea of how an exam question could look like. Number 6 for review. So according to Poiseuille's law, blood flow will increase if, which of these would be correct? Which of these would be correct? And the correct answer I would go with is choice letter D. Letter D is the correct answer. Only A and B are correct. C would not be correct because the viscosity of the blood, if that were to increase, the blood's getting sluggish, it's certainly not going to go up. It's going to go down. Okay? So definitely make sure to review this law. Very important when it comes to critical thinking skills. Moving on, we've looked at blood flow, and now we're going to focus on blood pressure. So this graph here is looking at blood pressure. The y-axis, blood pressure. So you can see the units here, millimeters of mercury. Units is always important in the medical field. And on the bottom, we're looking at, on the x-axis, the type of blood vessel. So when you record blood pressure, which you will do in lab, there's two pieces of data that you're going to be collecting. One is going to be what's known as the systolic blood pressure. And this is the pressure that's applied during left ventricular contraction and diastolic pressure. So that's the lowest level of arterial pressure. So typically the systolic pressure normally around 120 millimeters of mercury. But if you can have it between 110 and 120, even better. Diastolic pressure, typically that lower reading, so between 70 to 80 is a good range for someone who's healthy. So you can kind of see that here, and pressure's pretty high here, you can see overall. Especially blood pressure high in the aorta. And then eventually we look at arteries here, and then we eventually have a huge drop-off. And eventually you get to veins and the vena cavas, and blood pressure is extremely low. If you were to take these two values together, systolic pressure and diastolic pressure, you would end up with what's known as the mean arterial pressure, which is known as the MAP. So mean arterial pressure, this is the pressure that propels the blood to the tissues, the tissue cells of the body, and ultimately this declines with distance from the heart. We now look at, well, how do you take blood pressure? And again, you'll be doing this in lab eventually here, taking blood pressure. There's two things you're going to be using. One is the blood pressure cuff, known as the sphygmomanometer. So the sphygmomanometer here, the blood pressure cuff. That's always a fun one to say. So units again for pressure, millimeters of mercury for the sphygmomanometer, and you'll be using a stethoscope. You'll ultimately place the blood pressure cuff above the elbow here. You'll have the stethoscope, you'll see the pieces that go into your ears there. You'll have this in position, patient hopefully seated, and you'll slowly inflate the blood pressure cuff to around 200 millimeters of mercury. Again, it may vary from one person to the next. And you'll inflate this. There's a bulb right here. So you slowly will squeeze the bulb. That inflates the cuff with air. And you'll get it to that point, and then slowly you'll release the air. You have an air valve here. So you're slowly going to release the air, and you're looking for sounds. And this is ultimately going to be sounds that is the resultant of the brachial artery that you're listening to for constriction versus no constriction. So something important to consider here. Okay, so when you're listening closely, the first piece of data that you're going to collect is going to be the systolic pressure. Okay, so that's going to be the first sounds that occur as blood starts to spurt through that artery, in this case, the brachial artery. Then you're going to keep listening. And the second piece of data you're going to collect is the diastolic pressure. So those sounds are going to disappear then, and the artery then is no longer constricted, and blood then is going to be flowing freely. And it takes time to practice. If you've never done it before, believe me, it takes time. Believe me, I know. But very important skill to know. Many of you might already know how to do this. And if that's the case, it's practice. Another important thing to be aware of is, well, where can you take a pulse in someone? That's very critical. So ultimately, when you take a pulse, we're looking at overall rhythmic expansion, recoil of the arteries from heart contraction. And the pulse itself, the rate can change based off of differences in emotions and even the posture that the patient might be in. There are many different locations where a pulse can be recorded, as you can see on the diagram here. Some places are a little bit more easier than others. A very common location is the common carotid artery to find a pulse. I typically take my index finger, middle finger, and put it against the artery, and you can feel the movement of the blood. I'm actually doing it right now, and maybe you are too. And I'm still alive. The radial artery, that's kind of more of a common location too, that you can take a pulse if you needed to. I just found mine. And the thing is, these locations are also considered to be pressure points, which are key to go ahead and compress to slow or stop bleeding. Very important if you're, working in the ER, somebody comes in with a gunshot wound, okay? Blood's going everywhere. So one thing to consider is if blood is squirting all over the place, maybe a carotid artery was severed or something like that, okay, that's ultimately with blood squirting everywhere, typically that happens in an artery because of high blood pressure. If a vein was severed, you're not going to have blood squirting all over the place because the vein is under low blood pressure and the blood will actually pool in the vicinity. Another thing to consider is the fact that arteries typically are deeper in the body and veins are typically closer to the surface. Two very important terms when we think of blood pressure are the terms hypotension and hypertension. So hypotension and hypertension. Hypo, of course, is referring to low. So hypotension, low blood pressure. And that's usually where you have a systolic pressure reading less than 100. This is typically associated with long life, lack of cardiovascular disease. Hypertension, hyper referring to something elevated, high blood pressure. Considered to be a silent killer because it can lead to many different types of heart issues. And if this is sustained for a period of time, this is not good if it's greater than 140/90. We've all experienced high blood pressure short term, which is normal. Maybe you're fighting a fever. Maybe it's from working out. Maybe you got into an argument with someone. But then your blood pressure goes back down to normal. Risk factors for getting high blood pressure would be things such as obesity, so living that sedentary lifestyle, not eating good, so diet, poor diet, smoking, most definitely, and your genetics. The hypotension we just mentioned, how does someone end up with that? A big one is your genetics. Genetics. Okay, so we've looked at blood pressure quite a bit. And now we're going to take a look at factors which can aid in venous return to the heart. So these are factors that help with moving the blood back to the heart. So the first one we're going to take a look at here, number one, is the work of the respiratory pump. The respiratory pump. So this occurs when there's overall changes in pressure which are created during breathing, when you inhale and exhale, that ultimately move the blood towards the heart. As squeezing of abdominal veins, as thoracic veins expand. So it's all about breathing here, changes in breathing, that you're using the respiratory pump. Number two, another factor, vasoconstriction. Vasoconstriction. So vasoconstriction is when that vein is being compressed, typically under sympathetic control, when you're doing stress-related activities, when you're exercising and working out. Number three, the skeletal muscular pump. The skeletal muscular pump. This is referring to the idea that you have contraction of skeletal muscles, maybe in your legs as an example, that help to move the blood towards the heart. And there's valves that play a role here too. So this is being utilized all the time when you're lifting weights in the gym, you're working out. Okay, so this slide here is reviewing the skeletal muscular pump. You're utilizing this one, again, when you're exercising, when you're lifting weights in the gym. So when you're working out in the gym, maybe you're working out at Delta College's Fitness Center, or you're working out at the YMCA, or wherever you're working out. Maybe you're doing your leg machines, leg presses, whatever it is. And when you're doing that, the skeletal muscle is being contracted. And we can see that here on the diagram, right? We can see that here. That's what these arrows are representing. And when this is taking place, we take a look at our two valves here. So here's a valve that's open. The valve that's proximal to the area of contraction is forced open, important to propel the blood towards the heart. While the valve which is closed, distal to the area of the contraction, closes up. That's important so there isn't any type of backflowing of blood. So a very important concept to think about when you're working out and you're lifting your weights in the gym. We're now going to take a look at short-term controls of blood pressure. Short-term controls of blood pressure. And this is referring to where temporarily blood pressure increases or blood pressure decreases. And what plays a big role with this process are what are known as the baroreceptors. The baroreceptors. The baroreceptors are pressure-sensitive neuroreceptors. They're going to respond to changes in arterial blood pressure and also the overall blood vessel stretching. Baroreceptors are found in two major locations in the body. So they're found, we have the carotid sinuses. That's one location where we find the baroreceptors. So those are typically found along the internal carotid arteries. And the second location is the aortic arch. So right at the top of your aorta. So very close to a lot of those large arteries near the heart. So carotid sinuses and the aortic arch. Two major locations where we find the baroreceptors. And we go back to looking at the brain too, the cardiovascular centers, which we mentioned in the previous chapter. And they're going to be key for going ahead and receiving information, and ultimately there's a response to, in turn, a change in blood pressure that may occur, and even cardiac output and blood vessel diameter. So let's go ahead and take a look at this. So in our first scenario here, we look at an increase in blood pressure short term. So an increase in blood pressure short term. And we think of examples, perhaps, of things maybe in your daily life that might increase your blood pressure short term. So a great example that we'll go with is working out. You're doing exercise. Maybe you're going for a run. Okay? So you're going ahead and you're going for a run. You're running, right? You're excited. You're going for a run. Okay? You're at the gym. Maybe not the gym, but the track. And you're working out. So you're working out, and of course, when you start to exercise, right, your blood pressure is going to increase short term, right? Your heart rate goes up. The net result is cardiac output goes up. And when this happens, we look at ultimately how the brain's going to respond to this. All right, so first, right? So blood pressure goes up because you're working out in the gym. You're exercising. Messages then are eventually going to be sent to the baroreceptors in the carotid sinuses and aortic arch. So they become stimulated, these baroreceptors. Messages then are sent to your brain. In this case, going to the cardio-inhibitory center. There's going to be stimulation of the cardio-inhibitory center. And this is important because you need to get the blood pressure eventually down. So this scenario we're looking at here is you're exercising, right? You're on the treadmill, and you eventually get off the treadmill. Okay? Now it's a matter of getting back down to your rebound heart rate and the blood pressure getting back to a normal level. So the message is sent up to the brain from the baroreceptors. They stimulate the cardio-inhibitory center. So then messages sent away from the cardio-acceleratory center in your brain here eventually leads to a decrease in heart rate, decrease cardiac output, ultimately decrease in blood pressure, and you're back to normal. This happens after you exercise. Eventually, your blood pressure gets back to normal. What also can play a role is messages from the brain can also go to various blood vessels, okay? And this ultimately can lead to vasodilation of the blood vessels, and this leads to overall decrease in resistance. R here refers to resistance. So those are two big things that happen here. So let's run through this one more time. I know there's a lot of physiology here. I know it's a lot of fun, but we probably should go through this one more time. So let's say you go to your gym, right? You go to Planet Fitness or the Y or wherever, and you're happy, you're running, you're running 5 miles on the treadmill or 10 miles or whatever you're doing, and your blood pressure is going up, right? Heart rate's going up, cardiac output's going up, blood pressure's going up, and messages then are sent eventually to the baroreceptors, right? These baroreceptors become stimulated. Messages then are sent to your brain, to the cardio-inhibitory center. Inhibit. The word inhibit means to decrease. So, messages then from the cardio-inhibitory center can be sent to the heart, ultimately in turn can lead to decrease in cardiac output, decrease in heart rate. And messages can be sent to blood vessels. In this case, so vasodilation occurs, dilating, opening. This can lead to a decrease in resistance. Ultimately, blood pressure declines, gets back to a normal range. And that happens, of course, after you're done exercising. Okay? So definitely important to make sure to review this concept. We now look at the complete opposite scenario. So now we look at short-term changes in blood pressure, but now with a decrease in blood pressure. A decrease has occurred. So a great example here I like to use is, how about sleeping, right? We all like to sleep. We probably don't get enough sleep, but sleep. We like to sleep, right? So, yep, here you are, you're sleeping, right? And you're snoozing, maybe you're taking a nap or whatever the scenario might be. And when you sleep, of course, your blood pressure declines, right? So blood pressure goes down. Heart rate goes down. Cardiac output goes down. Messages then can be sent eventually to the baroreceptors, but this time they become what's known as inhibited. Messages are sent from the baroreceptors now to your brain, but now a new center is stimulated, and that's the stimulation of the cardio-acceleratory center. When you think of acceleratory, acceleration, increase. So, when you stimulate that center, ultimately, messages then are sent to the heart, and this can lead to an increase then in heart rate, HR, an increase in cardiac output. Messages also sent to blood vessels, and this can stimulate then vasoconstriction, causing resistance to increase. Remember, constriction, vasoconstriction. You have constriction of the blood vessel, like an artery. And ultimately, the blood pressure is going to go ahead and increase. Blood pressure goes up. Okay, so BP goes up. This is certainly the case when it's time to wake up, right? So you are sleeping, right? But eventually then your alarm goes off and it's time to get up, right? And it's 5 a.m. and you're not sure you want to wake up yet. But it's time to get up. You get up, you jump out of bed, right? And it's time to go ahead and the day has to start, right? And this is really the response which happens, right? So first, of course, you're sleeping, right? You're taking a snooze, and blood pressure falling, heart rate, cardiac output falling, messages sent to the baroreceptors, which become inhibited. They're going to send impulses to the brain, which stimulates the cardio-acceleratory center. And remember, acceleratory increase, right? Acceleration. Message is then sent away from the brain to the heart to increase heart rate and cardiac output. Message is also sent to blood vessels, and this leads to them undergoing vasoconstriction, increasing resistance, and ultimately increase in blood pressure. Literally, this sequence of events is taking place when the alarm goes off and it's time to get up to go to work, go to class. So definitely make sure to review how the baroreceptors work and looking at examples of what happens with the physiology when you have a short-term increase in blood pressure and a short-term decrease in blood pressure. This picture here ultimately is reviewing factors which cause an increase in mean arterial blood pressure. And again, a lot of these are probably no-brainers, right? So what can increase the mean arterial blood pressure? Ultimately, things such as exercise. We can see here exercise, so different stressors can definitely do that. And with that, of course, stroke volume heart rate leads to increased cardiac output to blood pressure. Of course, other things too, it could be some form of trauma or even increased body temperature could lead to this scenario happening too. We can see other things too that can definitely lead to the increase in resistance here. So body size can definitely play a role there. If somebody's obese or something like that. We can see other things here, dehydration, okay, high hematocrit, etc. Ultimately can lead to increase in blood pressure. And that's really the big thing to get out of this slide. We're now going to take a look at the different states of circulatory shock. The different states of overall circulatory shock. So when we look at circulation, looking at circulatory shock, any condition in which blood vessels are inadequately filled and blood cannot circulate normally. Number one, first example, hypovolemic shock. This is the result, large-scale loss of blood. So when someone loses a lot of blood, they can get into hypovolemic shock. So maybe it's a gunshot wound, maybe it's a fall, you hit a tree or whatever, accident with the chainsaw, maybe you nearly cut off your finger or your leg. So definitely not a good situation. Number two, vascular. This form of shock is typically taking place when someone has a severe allergic reaction. Maybe they're allergic to food like seafood or peanuts or something, or maybe they're stung by multiple bees, and so they end up with extreme vasodilation. This picture here kind of shows that. We have extreme vasodilation to the face here with this individual. So you have a big, the circulation is poor due to abnormal expansion. And number three here, cardiogenic. So overall pump failure of the heart. That's the case, of course, with the heart attack. So definitely make sure to know these. We now take a look at distribution of blood flow at rest and during exercise. Somebody's doing cardio. All right, so this diagram is looking at different locations in the body where we look at total blood flow at rest in comparison to total blood flow during the exercise, the cardio. All right, so at rest, when you're chilling at home, you're relaxing on the couch. When it comes to blood flow distribution, typically there's going to be more blood flow that's going to be in the urinary system and definitely in the abdominal area when we think of the GI tract. Those are two major areas where there's definitely going to be a lot more blood flow at rest. But when someone's shooting some hoops, okay, and exercise and doing some form of cardio, maybe somebody's going for a swim, or again, maybe somebody's going for their little run. I like to run a lot, if you haven't noticed with these examples. And it's time to go run, right? And maybe you're going to go out and run 20 miles for the day just because you love to run. You're doing the cardio. There's going to be certain areas of the body that are going to receive more blood flow. So that's going to be the heart here when you're exercising. You can see the big difference here. So heart, a lot more blood going through the heart. You're going to have a lot more blood ultimately that's going to be going to skeletal muscles especially when you're running, more blood flow going to muscles in your legs for sure, and your arms. And there'll be more blood flow going through the layers of skin too. There's going to be a decrease in the amount of blood flow to your GI tract and kidneys. Okay? More blood flow distributed here. But no matter what, the amount of blood flow going to the brain, you can see here, typically stays about the same. Of course, there's always situations out there, but normally that's going to be a constant. Okay? But typically less blood flow to the kidneys, urinary system, and less to the GI tract for that period of time. But of course, then when you stop exercising, well, then of course, fun things can happen. Hopefully that porta potty's nearby. Yeah. Okay, another thing we're going to take a look at here today is looking at the inverse relationship between blood flow velocity versus the total cross-sectional area in blood vessels. So we have two graphs here we're going to look at here. This top graph is looking at the total area of the vascular bed in comparison to many blood vessels throughout the body. So when we look at the overall total area of the vascular bed, the largest area typically is going to be the capillaries, represented here. Smallest overall area of the vascular bed, cross-sectional area, is the aorta. So pretty low here. And we can see that even with this image. This is supposed to represent capillaries right here. And then the below graph, we're looking at the velocity of blood flow versus, again, examples of different blood vessels in the body. So with velocity of blood flow, how fast is that blood moving? Ultimately, the fastest location would be the aorta. Okay, you have really high blood pressure. So right up here. The slowest area by far, the capillaries. So velocity really slows down in the capillaries. And you may be asking yourselves, why is that the case? Why is that the case? And the reason for that, why is the velocity of blood flow decrease significantly in the capillaries? Well, it allows for enough time for gas exchange to occur. It allows for enough time for gas exchange to occur. That's pretty important. We really need the blood flow to slow down so we get oxygen to tissues and CO2, carbon dioxide, removed from the tissues back into the blood. And likewise, it allows for enough time for exchanging of nutrients and hormones and getting rid of waste products. You need the blood flow to go very slow in the capillaries. All right, so now we're going to go ahead and review. So a couple review slides for us. So number 7 for review. You decide to run the Outer Banks Half Marathon in North Carolina this fall. Now, that's a fun one. You're running right along the Atlantic Ocean, and that's always a good time. When you are running, blood flow to the blank remains constant, while blood flow to the blank increases. So what would be the correct answer here? And the correct answer, hopefully you went with it, would be choice letter D. So letter D would be the correct answer for this question. All right, number 8 for review. You decide to take a nap in the afternoon, which leads to a drop in your blood pressure. But eventually now, maybe someone's tapping you to wake up, and maybe you fall out of your resting place. Messages are sent from your baroreceptors to your brain, to the blank, which leads to an increase in heart rate and blank of your blood vessels. So, what would be the correct answer here based off these choices? What would you go with here? What would be the correct answer here? So we'll give you a little bit of time here to consider what answer to go with. And the correct answer, hopefully you decide to go with this one. I would probably go with choice letter B for this one here. So it's the cardio-acceleratory center, because that's important. We got to get the heart rate increased, cardiac output, and of course blood pressure back to normal. And vasoconstriction of blood vessels. That's going to help, of course, to increase your BP back to a normal range. So that's an important one as well. We're now going to take a look at the capillaries in a little bit more depth. And remember, we talked about capillaries. We mentioned that you have oxygen-rich blood, which enters into the capillary. So oxygen-rich blood here on this arterial end of the capillary, okay? And ultimately, the oxygen goes out and goes to tissue cells. So that way you get oxygen in the tissue cells. And then in return, you have a buildup of carbon dioxide gas that's going to go out and go onto the venous end of the capillary. So you have an arterial end of the capillary and a venous end of the capillary. So the arterial end. The arterial end ultimately is transporting the oxygen like I've shown you, but also don't forget hormones and nutrients. And ultimately all of these items go to the tissue cells that are nearby. We also have the venous end. Here, carbon dioxide gas and metabolic waste products are going to be transferred from the tissue cells ultimately back into the capillary on the venous end. And then these items will eventually go out of the capillary into the venules and continue on through blood supply till they're eliminated from the body. So important to be aware of that, the arterial end and the venous end. All right, and now going on to our We take a look now at what's known as capillary filtration. So we've already been looking at blood going through a capillary from the arterial end to the venous end, and looking at movement of gases and hormones and metabolic waste products. But we now take a look at really movement of fluid now. So capillary filtration, fluid movements, and looking at the flow. So when it comes to the movement of fluid out of a capillary, ultimately we look at this, that it really depends upon two opposing forces that play a role with the movement of the fluid out of a capillary. The first type of pressure is known as the hydrostatic pressure. Hydrostatic pressure. And so hydrostatic pressure, this is the pressure that we can find internally in a capillary, and we can also find this hydrostatic pressure out in interstitial fluid. So interstitial fluid, all of this area over here, so all throughout here, interstitial fluid, really the area found outside of a capillary, because right here would be the capillary itself. The other type of pressure that we're going to talk about is what's known as the colloid osmotic pressure. Colloid osmotic pressure. So this is going to be the pressure we find, again, either within the capillary or interstitial fluid. So approximately about 20 liters of fluid are filtered out of the capillaries per day. And 17 liters of the fluid are going to go ahead and be reabsorbed back into the capillary. But 3 liters of the fluid are actually going to be transferred on to the lymphatic system, which is important because who knows what's in the fluid? There very well could be maybe viruses or bacteria or, heaven forbid, cancer cells. So it will go on to the lymphatic system. And ultimately, as we'll learn, this system is very important, associated with the immune system response to go after these foreign invaders. And this picture shows, ultimately, if you have a lot of hydrostatic pressure built up, that ultimately that can force out small solutes and even molecules, water molecules as a whole. When we look at the net filtration pressure, ultimately it's the movement of the fluid out of the capillary and some of the fluid coming back into the capillary. So a very important concept here, net filtration pressure, sometimes abbreviated as the NFP. So this is the formula that you use when it comes to looking at the net filtration pressure of the capillary. So the NFP, this is equal to the HPC, and this is referring to the hydrostatic pressure in the capillary. So capillary hydrostatic pressure minus the HPV, which is referring to the interstitial fluid hydrostatic pressure, OPC, and this is referring to the colloid osmotic pressure found in the capillary, and then we have OPIF, which is referring to the osmotic pressure found in interstitial fluid. So ultimately, you get an idea of what direction the fluid is moving based off your answer. Is the fluid coming out of the capillary? Is the fluid going back into the capillary? So this formula is very important, and we'll be taking a look at this very shortly here. So this slide here ultimately gives us an understanding of what exactly is the net filtration pressure and fluid movement in the capillary. Here you can see the formula being put to use. So again, when we look at a capillary, we have the arterial end on this side. So we have the oxygen-rich blood on this side of the capillary. And then the other side of the capillary over here, we have oxygen-poor blood. So first on the arterial end, ultimately, when we look at the arterial end, the pressure that's going to be the dominating pressure here is going to be that hydrostatic pressure. So this is pressure that's actually building internally in the capillary, which will actually force the fluid out. So important to remember, when we look at the arterial end of the capillary, it's the hydrostatic pressure, it's the dominating pressure. And typically, when you go ahead and calculate your answer, you'll typically end up with a positive reading. So here you can see the NFP, you have a positive 10 millimeters of mercury, and the fluid is going to go ahead and move out of the capillary, okay? Moving out of the capillary. So ultimately this direction, enter interstitial fluid. And then we look at the other end of the capillary. Of course, like I've mentioned already, some of that fluid continues on to the lymphatic system, but you do have some fluid, again, that comes back into the capillary. So looking at the venous end of the capillary, the major pressure here is going to be that colloid osmotic pressure. And this is kind of a sucking pressure that's actually going to be sucking that fluid back into the capillary. Okay, so arterial end, you have the movement of the fluid going out here to interstitial fluid. And then on this end over here, the venous end here, you're going to have the fluid that's being sucked back into the capillary here on the venous end. So the dominating pressure on the venous end is going to be that colloid osmotic pressure. And that's typically reflected, you usually have a negative number here. So typically a negative number for the value here. So usually a negative number is what you typically get, reflecting that the fluid is going to be pumped back into the capillary. Okay, so a wrap up here again. On the arterial end of the capillary, the hydrostatic pressure is the dominating pressure here. That's going to force the fluid out of the capillary into interstitial fluid. And you typically end up with a positive reading that's reflecting that. Then when we look at the venous end of the capillary, the colloid osmotic pressure is the dominating pressure here. And this is a sucking pressure that's going to go ahead and suck the fluid back into the capillary here. And again, you typically end up with a negative reading here that's showing that. So you might be asking yourself, so what's the entire purpose? What's the purpose of even learning this? Well, it ultimately shows you, do you have normal movement of fluid in your body? So if these values are off, who knows? You might end up with some type of maybe lymphedema, edema fluid buildup in your body, where maybe you're getting possibly too much fluid, maybe going into your lymphatic system, maybe you're not getting enough. So ultimately, it's a proper reflection of normal movement of fluid. So it's kind of the importance of looking at this and reviewing this. This is just another picture here, again, showing the net filtration pressure of the capillary. I took this one right out of your textbook. So this is another definitely important slide to review and look at as well. And you can also see here, like I mentioned before, where we have this positive reading here of the NFP. Again, this is the arterial end here. And then over here, you could see a negative reading. Again, the colloid osmotic pressure dominates here. You have a negative reading reflecting that the fluid is coming back, reabsorption of the fluid. Okay, so arterial end, hydrostatic pressure is the dominating pressure, forcing the fluid out of the capillary. So again, we can see that here, and filtration, and here reabsorption. All right, so the last couple slides, we went over the concept of calculating the net filtration pressure of the capillary. We looked at several diagrams. But now it's your turn to go ahead and look at calculating this overall important process of movement of fluid out of the capillary. So in your course packet for Bio 153, if you look under lecture handouts, you'll see several handouts that refer to this process of calculating the net filtration pressure out of the capillary. And you know what those handouts are going to be. You'll see like a cylinder type of cube. And I'm going to go through the first problem with you, and then I'd like you to work on the second problem set at home. So the first one we're going to do here, and hopefully you have this in front of you. So problem set number 1. So number 1, the figure above shows a capillary leading from an arteriole to a venule. The hydrostatic pressure of the blood is 50 millimeters of mercury at the arterial end and 15 millimeters of mercury at the venous end. The hydrostatic pressure of the interstitial fluid is 0 millimeters of mercury at both ends. The osmotic pressure of the blood is 30 millimeters of mercury, and the osmotic pressure of the interstitial fluid is 5 millimeters of mercury along the entire capillary. Please calculate the net filtration pressure at the arterial end and venous end. So the first thing we're going to do is go ahead and have our equation here for us so we have a nice visual. So the NFP, equals the HPC, so HPC minus the HPIF in parentheses, minus, in parentheses, OPC minus OPIF in parentheses. Okay, so again, what do these terms mean, right? Of course, NFP, net filtration pressure, referring to the capillary, so that's equal in parentheses to the HPC, so the hydrostatic pressure found within the capillary minus the HPIF, hydrostatic pressure in interstitial fluid, minus, in parentheses, the OPC, so this is referring to the osmotic pressure in the capillary, minus the osmotic pressure in interstitial fluid. So first, how about we will go ahead and calculate the arterial end. So the arterial end, that makes sense. So we think of the overall order of movement of the blood. So we'll do the arterial end first, and then we'll calculate the venous end. So sounds like a good plan, I think. All right, so the arterial end All right, so based off of our statement, now it's a matter of filling in the values. So NFP is equal to, we'll put in here, we have 50 minus 0. And again, the 50 is a reflection of the hydrostatic pressure in the capillary. 0 is a reflection of the hydrostatic pressure in interstitial fluid. Minus, in parentheses, 30 minus 5. 30, a reflection of the the overall colloid osmotic pressure in the capillary, and 5, a reflection of the colloid osmotic pressure in interstitial fluid. So 50 minus 0 is 50. 30 minus 5, 25. And then 50 minus 25, your final answer should be positive 25 millimeters of mercury. And referring to the effect that the fluid moves out of the capillary bed. And this is also a reflection here, when you get this answer, that it's the hydrostatic pressure that's the dominating pressure. So the hydrostatic pressure is going to dominate here. Okay? So hydrostatic pressure is going to dominate. Okay, so let's go ahead and take a look now at the venous end. So hopefully you've had the opportunity to write all this down, but I want to make sure there's plenty of room here. So we're going to go ahead and erase all this now. All right, we're almost there. Looks pretty good. Chalkboard looks wonderful. Marker board, right? All right, now we're going to focus on the venous end. So, focusing now on the venous end. All right, so venous end. So based off of the values here, we can, and the information in our paragraph, we'll find that NFP is equal to 15 minus 0 in parentheses, minus, now in parentheses, 30 minus 5. So we go ahead and 15 minus 0 is 15. 30 minus 5 is 25. And again, you might be wondering, well, how do we get these numbers, Chris? Well, 15 is referring to the hydrostatic pressure in the capillary. 0 is referring to the hydrostatic pressure in interstitial fluid. 30 is referring to the colloid osmotic pressure found in the capillary. 5 is referring to the colloid osmotic pressure in interstitial fluid. So again, we have 15 minus 25 here. So we can work on making this a little clearer here. So 15 minus 25. So when we do that, 15 minus 25, ultimately we get an answer of negative 10 millimeters of mercury, referring to the effect that the fluid moves into the capillary bed. So that's your final answer here. If you get a negative value, that's typically a reflection of the fluid being sucked back into the capillary. On the venous end, of course. And the pressure that dominates here, it's a reflection that the osmotic pressure dominates. All right, so now I'd like you to work on the second problem set at home. And so we'll give you a couple minutes here, and then I'll go ahead and give you the answers. Okay, so go ahead and work on the second problem set. And again, it's really important to make sure you know how to do this, because you never know, there might be a question like this on your exam. And you'll have more practice in the homework for chapter 21 as well. So go ahead and take some time to go ahead and work on this next problem set. All right, so hopefully you have the opportunity to go ahead and work on that. And I'm going to go ahead and erase all of this here. And let's see if you got the correct answers here. All right, so let's go ahead and first see what you got for an answer for the arterial end. So for the arterial end, so based off the data that we have here, based off the information provided for the arterial end, you would have had 60 minus 0 in parentheses, minus in parentheses, 35 minus 3, and then 60 minus 0 is 60. 35 minus 3 is 32. And that should equal a positive 28 millimeters of mercury should be your answer. And again, this is a reflection that the fluid moves out of the capillary bed. So hopefully you got that as an answer here. And of course, the dominating pressure here again would be the hydrostatic pressure. Let's take a look now at the venous end. Let's see what type of value you got here. So the venous end. So NFP would be equal to 20 minus 0 in parentheses minus in parentheses 35 minus 3. Doing the math here, you should end up with 20 minus 32. And your final answer here should be negative 12 millimeters of mercury. And again, this is a reflection that the fluid is going to move into, so the fluid is going to go ahead and move into the capillary bed. And we'll fix this here. You've probably realized, oh, Chris has wonderful handwriting. All right. So, all right. And here we have capillary. Okay. So, again, this is the correct answer. Negative 12 millimeters of mercury. Fluid is going to go ahead and move into the capillary bed. And this is because the colloid osmotic pressure is the dominating pressure on this end of the capillary. It's the sucking pressure that sucks some of that fluid back into the capillary itself. So hopefully you've got these answers here. Remember that you do have answer keys in your course packet too. And remember, you will have a question like this on your exam. So definitely very critical to review this and know this. So I hope you've enjoyed the blood vessel physiology. And until next time,